and what we'll do is we'll finish the discussion of gametogenesis in females, oogenesis, and we'll take a look at what happens to that follicle in the ovary as the egg develops. And then we'll also take a really quick look at the uterine cycle, and then we'll also look at fertilization as well. And then that will kind of finish things up. So a couple of small things. We have a couple of microscope slides to look at and things like that, but it should be pretty painless. I don't know what you heard from people in the other lab, but it should be pretty painless. So let's go ahead and just kind of dive right in. Let me bring up the. Thought I turned the projector on. There we go. All right. So our female reproductive models. We have a number of different female reproductive tract models kind of scattered about. They, like the male models, all have their strengths and their weaknesses. So, I would definitely take the time to make sure that you're looking at all of them to see what they can all show you. All of them, though, are some variation on this theme where there's this sort of sagittal section, but you can also sort of look three-dimensionally at it. So that's essentially what we have right here. So I just kind of wanted to go through a couple of the major structures, a couple of the nuances of these, and a couple of other models, and then I'll let you guys work for a little while. So one of the things that we see is when we hold the model, and this is easier to do obviously when it's right in front of you. You hold the model like this, you can obviously see inside off of the mid-sagittal line, and you can see your ovary right here. You can see that ovarian ligament right here that connects the ovary to the uterus, which is this structure. The fallopian tube that we talked about earlier today is right here, leading to the actual uterus itself. And then the other thing that you can see, sort of, and this is kind of one of the nuances I wanted to talk about, is the ligaments that are here. So the ovarian ligament is right here. This connects the ovary to the actual uterus. The round ligament is right here. But the other two major ligaments that help to support the female reproductive structures are the suspensory ligament and the broad ligament. The suspensory ligament comes off of this side of the ovary because it, as the name implies, it suspends the ovary within the abdominal pelvic cavity. These models don't show it terribly well. They have it kind of built in with these blood vessels that are going up right here, so they don't show that suspensory ligament terribly well. I have another model that I'll show you in just a second that shows the suspensory ligament a little bit better, but it would be embedded within these blood vessels that we see right here. Okay. The other part of this, of course, is the broad ligament, which isn't cord-like, right? Like the round ligament, the ovarian ligament, even the suspensory ligament are all cord-like ligaments. But the broad ligament is a big sheet of connective tissue. And so it's all through here. So like the broad ligament is here, the broad ligament is here, the broad ligament is here as well. So it's kind of spread throughout. They don't really do more than just kind of assume that you know it's there, I guess. There's not like a structure they put in there that really allows you to see it. But this is where you would find broad ligament here, you find it here, and as far as the uterus itself goes, we can see that it is a thick muscular organ. You have three layers. You have the outer parametrium. You have that layer of smooth muscle, the myometrium, and you can see just how muscular it is. And then you have that inner endometrium that goes around the inside of the uterus. So this model does a pretty good job of showing us all three layers of the uterus. Cervix is right here, and then of course we have the vaginal canal right here, leading out of the body. Here you have the labia minora and the labia majora. flora, we said this morning, that those are homologous to the scrotum. And then you have the clitoris right here, which is homologous to the penis. So this model does a pretty good job of showing the major structures of the female reproductive tract. But again, there's a couple of things that are sort of challenging. Let's start with that suspensory ligament that we have right here. I have a better model for that. And it's sort of an unexpected place, but this model right here is a little bit better for showing us that. So the purpose of this model is to show the different stages of follicular development in the ovary. This is an ovary right here. The purpose is to show the different stages of follicular development. We'll talk about the stages of follicular development later today. So we'll come back to this model specifically regarding the different follicles that you see in here. But we do have a little section of ovary right here. And then you have your fallopian tube right up here as well. So you have those fimbriae of the fallopian tube, and you have the infundibulum, the ampulla, and the isthmus of the fallopian tube. So every part of the fallopian tube is shown here, and they actually do a pretty good job. This is really hard to see on these models because it's so small, but on this model it's blown up and it's actually pretty good. But additionally, we take our fallopian tube off. Additionally, you have broad ligament right here. This is that sheet-like broad ligament that we have, plus ovarian ligament. And then on this side, you have suspensory ligament. And this model actually shows the suspensory ligament really, really well, whereas some of the others don't necessarily show it, right? So I think they do a pretty good job with this model to kind of show the different parts. So our fallopian tube kind of goes on like that. So again, broad ligament, ovarian ligament, and way over here on the lateral side is the suspensory. Again, we'll kind of come back to this model a little bit later on today for some of the internal structures here, but this one does a pretty good job kind of showing it. Called the greater vestibular gland. Remember how the bulbourethral gland that we looked at on Wednesday was just off of the mid-sagittal line, so not every model showed it. The greater vestibular gland is kind of the same. What it does is it has a secretion that provides lubrication for the vagina for intercourse, and it's kind of right here, right at the entrance to the vagina. But the model doesn't show it because the model is showing you everything that's here on the mid-sagittal line. So what we have to do is we have to look around just a little bit for it. And this model actually shows it fairly well. So again, we're looking at this guy like this. This is the vagina right here. That vestibular gland would be right here. This is not it. This is a magnet. But if we turn the model over a little bit, we see that greater vestibular gland is kind of hiding right here underneath. We can see it from the inferior view, I guess. Let me zoom in just a little bit. We can see that greater vestibular gland is right here. It's the one labeled number 23. So it's right at the entrance to the vagina right here. And obviously there would be a duct kind of going over here for that secretion to kind of make its way. But we just can see it right here. Unfortunately, they don't really show it right here because, again, we're on the mid-sagittal line. So that greater vestibular gland is going to be this guy kind of hiding right here. So a couple of nuances to this. A couple of things to sort of pay attention to. Let's go ahead and take a quick look at your lab. There's a couple of aspects here. I think it's helpful to make comparisons between the two processes, between spermatogenesis and oogenesis in females. One of the things that we said is that not a whole lot happens during embryonic development in males. It's really not until we reach puberty or adulthood, as it were, that we actually enter the process of spermatogenesis. So really not much happens in males until puberty actually starts. And the other side of it is that with those stem cells, we see that not all of them are going to enter meiosis. Part of them are going to stay in the seminal vesicles, or I'm sorry, seminiferous tubules, and they're going to continue to undergo mitosis. In females, what we see is we see a lot more happening during embryonic development, right? We see that the mitosis of those oogonia, those stem cells, has completed, and we see that all of those oogonia are going to enter meiosis. And they essentially are kind of frozen in time during that, after birth until puberty starts, right? So here we see these primary oocytes. They've started their first meiotic division, and we see that really then nothing is going to happen until puberty. So, obviously depending on the individual, there's anywhere from like a 10 to 14 year period where nothing really happens, right? And then after that, we see that they continue to go through their meiotic divisions, but they don't all go at once, right? I mean, with males, as soon as something enters meiosis, it just develops into a sperm. But here we see that we don't have that happening. Numbers vary, but I think I've seen the most common number I've seen is that a woman is born with approximately 20,000 of these primary oocytes in each of her ovaries. So that's a pretty significant amount, right? If you did a little bit of math and you figured that a woman has cycles for approximately 50 years, right? So if we go from puberty starting at age 10 to menopause at around age 60, that's a 50-year time span. If she has a cycle with one egg per month for, what did I say, 50 years, that's 12 times 50, I think that comes out to 600, right? So 600 eggs are what she would need in her lifetime. She has around 40,000, so there's still a huge number there, but it's not an infinite number either, right? So we have to kind of remember that. But nevertheless, this starts before birth, and then it's kind of frozen in time. And then once per month during the ovarian cycle, which is what we want to talk about today, one of those secondary oocytes is going to continue on with its meiotic divisions. Another key thing to remember is that in spermatogenesis, we end up with 4 sperm, right? That's what meiosis tells us, is that we start with 1 diploid cell, and we get 4 haploid cells as a result of that. But in oogenesis, we actually only get 1 egg, right? So why is that? Well, we still have the same meiotic divisions, but we see that the second cell degenerates as what we call polar bodies. So here at the end of the first meiotic division, we see that polar body degenerating. That's what would become 2 of the 4 cells, but it actually degenerates. The other one degenerates months later, and actually not until after the egg has been fertilized. Because that's another key point, is that meiosis in oogenesis does not finish unless the egg is actually fertilized. Okay? And that's when meiosis actually finishes, and that second polar body is kicked out, and then we have a complete process. So it's done in a fairly regulated environment, as opposed to spermatogenesis, which is not regulated very much at all. So what we wanted to do today is we wanted to take a look at these structures. But what's important to understand is that these oocytes are going through their meiotic divisions, and around them we see that there's this thing called a follicle that's developing. Okay? So those primary oocytes that a woman has at birth in her ovaries are suspended on what we call primary follicles, right? Which is just a little capsule of connective of epithelial tissue. And then what happens is when one of these goes through its cycle, when it goes through development, what happens is that follicle is going to grow and grow and grow around it until that follicle eventually ruptures and the egg is essentially kicked out of the ovary via a process called ovulation. Okay, so let's go ahead and take a look at this. So this diagram kind of is showing a lot. There's a lot in here. So let's go ahead and kind of go through this really quickly. So here we see the ovarian cycle on this frame. Down below we have a timeline. This is on average the ovarian cycle is a 28-day cycle, and you know obviously there's a little bit of variation with that, but on average it's a 28-day cycle. So at the very beginning we see that primary follicle, and we see that it grows and grows and grows until it eventually becomes a mature follicle that we see right here. These mature follicles are sometimes called Graafian follicles. Graafian or mature follicle. You'll notice there's a lot of empty space in there. That empty space is called the antrum of the follicle, and we'll be able to see that today. And that is what ruptures, and we see that the egg is ovulated. And in that 28-day cycle, the ovulation occurs approximately on day 14, so about halfway through the cycle is when it occurs. Ovulation is triggered. You'll notice in the frame above the one we were just looking at, ovulation is triggered by both by a spike in the gonadotropins, but luteinizing hormone is really the culprit there. So luteinizing hormone triggers ovulation to occur. Shut this really quick. Okay, so that's what we see here. We see the luteinizing hormone spikes, and then we see the ovulation occurs as a result of that. And then what happens is all of that tissue that has gone into making that follicle starts to degenerate. It starts out as a structure that we call the corpus luteum, and then eventually it degenerates into a structure called the corpus albicans. And both of these essentially are scar tissue. We'll talk a little bit more about their importance here in just a minute. So when we look at these models, a couple of our models show the different stages of follicular development. Let's go back over here really quick. One of them is this model of the ovary that you see right here. Zoom out just a little bit. This model shows, your primary follicles right here. Again, you're going to have several thousand of these per ovary. And then once per cycle, we see that one of these is going to grow into secondary follicles, eventually turning into a Graafian follicle with that large open space called the antrum. Here's your egg right here that's actually being ovulated. You see that follicle has ruptured, ovulation has occurred, and then we have the corpus luteum, which is the beginning of that scar tissue. Corpus luteum literally means yellow body, so this is a corpus luteum, and then we have the corpus albicans examples here. Albicans means white, so these are white bodies, and those are our scar tissue that sort of come out of this. And we'll have a chance to see all of these under the microscope later today. This model also shows the different phases of follicular development. I like that this one actually does it in order. So we go from our primary to our secondary to our Graafian to a ruptured follicle, and then a couple of examples of corpus lutea, and then a corpus albicans right here. So we can kind of see those different things. So these models show these different phases of follicular development really well. And we know that the egg inside of those follicles are undergoing those meiotic divisions. Right? So that's the ovarian cycle, and it's about a 28-day long cycle. The other cycle that has to coincide with the ovarian cycle, of course, is the uterine cycle or the menstrual cycle, right? Because remember, the point of the ovarian
ovarian cycle is to release an egg that will hopefully be fertilized, right? If fertilization occurs, we need to have a place for implantation to occur and for the baby to grow, right? Or more specifically, for the placenta to grow, so that the baby can be nourished. And that, of course, is the uterus. So at the same time that this is happening, there's a second separate cycle that's happening in the uterus, in which the endothelial lining, the endometrial lining of the uterus, is proliferating. Those epithelial cells are proliferating. The underlying connective tissue has blood vessels that are going to be growing into it, and this is going to provide a place for implantation to occur. So you see a thickening of the endometrial lining during the uterine cycle. In order to prevent that endometrial lining from sloughing off during menses, we have to have something that tells the menstrual cycle from essentially to stop. That signal that tells it to stop is the hormone progesterone. Okay, so in the days after ovulation, the corpus luteum is going to start to secrete massive amounts of progesterone. We see that progesterone levels here are rising considerably in the days after ovulation because the corpus luteum is secreting that. Okay? What that's going to do is it's going to prevent this endometrial lining from being sloughed off. The corpus luteum doesn't last for more than about 10 days or so, though. And then what would happen if pregnancy had occurred is that the placenta would have established itself by then, and the placenta would be making its own progesterone, and it would keep the endometrial lining in place. Okay? However, if pregnancy does not occur, after the corpus luteum degenerates and the corpus albicans becomes more dominant, you see that progesterone levels drop, and that triggers the onset of menses, which is the beginning of the next cycle. Okay? So that endometrial lining from the previous cycle is sloughed off during menses. Lasts, somewhere between 5 and 7-ish days or so. And then the endometrial lining starts to build itself back up again in preparation for a potential pregnancy. Okay? And again, if the cycle passes again without pregnancy, as soon as progesterone levels drop, it triggers the onset of menses, and we see that the cycle starts itself over again. So we have things happening in the ovaries, but then we also have the uterus sort of prepping itself for potential implantation of an embryo. A couple of other things about this graph. You'll notice that the very bottom frame here is basal body temperature. Kind of an interesting thing, you'll notice that when ovulation occurs on day 14 of the cycle, you'll notice that there is a very predictable and reliable rise in body temperature of about 0.3 degree. That's not a lot, but it is predictable and reliable. Some women do choose to track their cycles via that change in body temperature. They sell special thermometers that can measure body temperature down to 0.1 degree. But man, it's really hard. You have to do it literally at the exact same time every single day. Like if you're 2 minutes late, you've missed the boat. You have to do it at the exact same time every day. You have to do it before you've moved out of bed. So like you wake up, you put the thermometer in your mouth and you measure your temperature. You can't talk, you can't get up and go to the bathroom. Any of those things will raise your basal body temperature and you'll have missed the boat. So it's a really challenging thing. Women who choose to track their cycles this way have to be very regimented in terms of doing it. But women who are regimented about it get really good and reliable data from it because it's very predictable and very reliable. You just can't ever mess up or you completely screw up your whole data set. So it's something that if you choose to track a cycle that way, you have to be really, really strict about doing because it can be really challenging. So anyway, it's kind of an interesting thing. But at any rate, so we have that. It's a lot going on in the female reproductive system in order to make this work. But hopefully, reproductive system, maybe not you, but hopefully for the reproductive system, that egg ends up being ovulated. Or not ovulated, fertilized, sorry. Fertilization, skip around just a little bit here. Fertilization happens quite far up in the female reproductive tract. We see that most fertilization happens almost right after ovulation occurs. Okay, happens usually in the ampulla or the infundibulum of the fallopian tube. So, from a sperm's point of view, that's a long way to go. I mean, they have to make it from the end of the vagina, which is right down here, through the cervix, through the uterus, and all the way up the fallopian tube in order to be able to fertilize the egg. That's a really, really long way. Human ejaculate, I think, has, numbers vary, but I think roughly 2 million sperm per ejaculate is what you're looking at probably. Only about 50 of those 2 million sperm actually make it to the egg. So, like, we're talking about a massive die-off, like a 99% plus die-off of sperm. You don't even make it to the egg. Right? So it's a long swim and a long journey. The egg needs to be fertilized within about 24 hours of ovulation or it won't be viable. But sperm can actually hang out in the female reproductive tract for up to 4 or 5 days and stay viable. So there is a little bit of a window there that can be used. The egg does present a couple of barriers to ovulation. So I have a note in my notes, but also here, who put this menu bar up top? Okay. There are a couple of barriers to ovulation. So this shows the egg that's actually ovulated right here. This model actually does a really good job. So this is the ovum or the egg. This is where you would find your DNA. There are 2 primary barriers here. Is this acellular layer that we see surrounding it. This is a layer of glycoproteins called the zona pellucida. And then the outer one is the cellular layer that we see right here called the corona radiata, which literally means radiating crown, right? So both of these barriers are in place and make it a little harder for the sperm to fertilize the egg. You guys will remember on Wednesday when we looked at the sperm, we saw that the head of the sperm has that little cap on it. You guys remember what that was called? It's called the cap was called the acrosome. Right? So that acrosome has enzymes in it that allow the sperm to penetrate these layers. Okay? So in order to get through these layers, there needs to be something called the acrosomal reaction, in which the sperm's enzymes are actually going to be able to dissolve these layers. Right? So the first sperm to sort of get to the egg here gets to dissolve these layers, and then it gets to combine its DNA with the DNA that's in this nucleus, and then we have a diploid cell that can undergo mitosis and development and grow into a new individual. Right? The egg also has a protective measure against what we call polyspermy, which would be more than one sperm fertilizing it. So the acrosome, the acrosomal reaction allows a sperm in here. But as soon as a sperm fertilizes the egg has a second chemical reaction called a cortical reaction. And what that does is it sends up a second barrier, and that is actually impenetrable by sperm. So it sends up a second barrier preventing any other sperm from fertilizing it. And the really cool thing about the cortical reaction is we can actually see it happening in real time. Let's go back over here really quickly. So I'm going to show you this. Let's see if we can find an example of one. Like some of them are a little bit bigger, like this guy right here, for example, is obviously a little bit bigger. Some of these are also big. So we do see some some secondary follicles in here as well, but the majority of what we see on the outside are all those primary follicles on the outside of our ovary. Okay? So lots and lots and lots of these primary follicles kind of all over out here on the side of our ovary, and you can see as you go around that tends to be where they're at. Again, some of these are a little bit bigger, and those are probably secondary follicles, but many primaries. Okay, but then what we are obviously here to look at are these Graafian follicles. And remember, those Graafian follicles have that massive open space called the antrum associated with them. So let me come over here and see if we can find some examples. There's a lot of them here, so there's no shortage. I'm just trying to find a good one. Just look for a minute here. Again, no shortage. I'm just seeing what I can find here. This is a good one here. So the Graafian follicle is going to be this structure. There's obviously a lot here to choose from, but this one is pretty good. I'll explain in a couple of minutes why I chose this particular one to use. But this structure is your Graafian follicle. So the follicle is this entire thing that we see right here. This entire thing is the follicle. Excuse me. That open space that we see in the middle of it, that's the antrum that's kind of going on around here like this, right? So all of this is the antrum that we see, okay? And then of course the egg itself is going to be right here in the middle. There's your egg, okay? The other thing that you can see here, and the reason I did choose this one, is because you'll notice here that you also have the zona pellucida right here. That's this acellular layer that the arrow is on, this kind of bluish line that we have right here. And then you also have the corona radiata that's right here as well. So this entire structure, I'll go ahead and put a white dotted line around it. When this thing is ovulated, it's this entire structure that's going to get ovulated. And those two things, the zona pellucida and the corona radiata, are going to provide those barriers to fertilization that the sperm has to get through in order to fertilize that egg. Okay? So let's take a minute on our own slide. Let's find a really good example of a Graafian follicle, one where you can identify that zona pellucida and that corona radiata. The follicle, after the follicle ruptures, right, after that Graafian follicle ruptures and the egg is ovulated, what we're going to see is that leftover follicular material, that leftover follicular tissue, is going to start to degenerate as this structure called the corpus luteum. And that's going to be what secretes progesterone until the placenta can secrete its own. So we're going to see that progesterone being secreted by the corpus luteum for about 10 days or so. So we're going to be looking at these next. Obviously, they don't look yellow under the microscope. They've been stained, little purple stains like everything else we looked at this semester. But just a quick note, notice that before they get very mature, they kind of look donut-shaped, like there's hollow space in the middle. That's that leftover antrum that's just shrinking and shrinking and shrinking. But they can also be solid in terms of their appearance as well. So we're going to see both types under the microscope. So I'm going to show you one first that's just solid. And I wish I had something more exciting than scar tissue to show you for your last slide, but it is what it is. So let's just go ahead and take a look at what we've got here. But let's take a look at some corpus luteum under the microscope. So again, you don't need to be any higher probably than low power. This is my ovary here. You'll notice that the ovary looks different than the one that we just looked at. This one has obviously moved on to a different phase. We're no longer in that follicular phase, as it's called. We're now in what's called the luteal phase.
of the cycle, and there are corpus luteum that are present. Ovulation has occurred, and now all those Graafian follicles that we saw in our previous slide are all now degenerating as corpus luteum. Right? So, for instance, this is a great example right here of a corpus luteum that we see right here. It's just a mass of scar tissue within the ovary. Right? So let's try to find a corpus luteum on our own slide.